So this would be versus autonomic. Okay. So when we talk about the somatic division of the nervous system, this includes motor neurons. So motor neurons innervate skeletal muscle. So in general, this is a conscious part of the nervous system. So remember that we talked about the cortex, and there was a somatosensory cortex that detected touch and also detected muscle stretch and position of your appendages. But then there was also a part of your cortex called the motor cortex. And so that sends action potentials out through your body, out through your central nervous system, and then into the peripheral nervous system through the motor neurons. And do you remember what part of the spinal cord do the motor neurons leave? Do they go the ventral route or the dorsal route? Does anybody remember? It's actually ventral route. And so the ventral route is the motor neurons coming out of the spinal cord. And there are some diseases where we get damage to those motor neurons in the spinal cord. And so an example of that is polio. So polio is actually caused by a virus that damages motor neuron cell bodies, and that is why it leaves you paralyzed. And so we get vaccinated for the polio virus in order to prevent having that horrible disease that causes us to end up in either dead or in wheelchairs. Okay.
What about multiple sclerosis?
Multiple sclerosis, we talked about that. That attacks what? Does anybody remember? Doesn't attack the neurons directly. It attacks the glial cells that are associated with the neurons. And what is the glial cell that's in the peripheral nervous system? That gives the axon its myelination. Schwann cells, right? So multiple sclerosis is not the motor neurons. The motor neurons stay fine. It's the myelination that gets removed, and then that causes all kinds of problems. Yep. Okay, so these are the motor neurons. So this is the nervous system, right? Okay. When we look at skeletal muscle, the muscles are not just composed of muscle tissue. So each muscle is a separate organ. So remember, in our biological hierarchy, we go from cells to tissues to organs. And what this means is that each muscle contains not only skeletal muscle tissue, but other types of tissues as well, including connective tissue. So for an example, connective tissue surrounds the skeletal muscle. And sometimes this is called the fascia. Right? That's F-A-S-C-I-A. So you talk about muscle fascia. And so one of the interesting things about the muscle fascia is that it is innervated by the nervous system. And it's actually believed that people that have a disease called fibromyalgia, it used to be believed that people that had that symptoms of just pain throughout their body, that it was just psychological. And so recently they've discovered that there are nerve nociceptors, pain receptors in the muscle fascia. And for some reason, those don't work very well in people that have fibromyalgia. And so that feeling of pain in the body is believed to be due to that muscle fascia. And so if you were a massage therapist, you would study the muscle fascia, right? Because what they want to do is they want to keep the connective tissue healthy around the muscles. And so they don't want the connective tissue starting to stick together. And so when the when the connective tissue starts to stick together, sometimes that's called adhesions, right? And so they try to, especially scar tissue, they try to massage that out and keep the connective tissue nice and healthy. Okay. So that actually surrounds the skeletal muscle. And then obviously we have nervous tissue, right? And we have blood vessels. So I'll put nervous tissue and blood vessels associated with each muscle. So this is why they are considered to be organs, the next level of that hierarchy. Yep, question?
Yeah, is the fascia what's responsible for muscle memory?
No.
What is that?
There is no memory in the muscle tissue itself. It is in the central nervous system. So muscle memory, we talked about cerebellum. So that ability to like walk, ability to play a musical instrument can be coordinated by the cerebellum, which is different than your cerebrum. So that's neuromuscular coordination. So there's really no such thing as
I don't know
in the
I
muscles.
read it somewhere, or I heard it something, but that the fascia is what's around the muscle and what makes it move, right?
No, it encapsulates the muscle, and the muscle tissue moves within the fascia. And so if that fascia doesn't stay loose, then the muscle can't move. And so that's why people think the massages are so good, is that keeps that muscle fascia nice and loose, right? And especially like I have surgery on my back, right? And that place where they cut into me is scar tissue, and so my muscle fascia is not great right there, right? So if they can massage that, and they actually tell you to massage scars because you don't want the connective tissue starting to adhere incorrectly. Yeah. Sometimes they call it the fuzz. The muscle fascia is what is called the fuzz. F-U-Z-Z. Okay. There's actually a YouTube video that talks about that fuzz, but it's not quite scientifically accurate enough for me to play it in class, but it's kind of interesting. Okay, so when we look at the skeletal muscle, we have to talk about the different terminology, and I have a handout here. So you need to know these different structures in the skeletal muscle. And so when we look at muscle, there's two root words that oftentimes come up, and one is myo. So M-Y-O means muscle. And then sarco also means muscle, because sarco means flesh. So myo and sarco, whenever you see those root words, you know that that has something to do with the muscle tissue. Okay. So when we look at the skeletal muscle itself, we can talk about muscle fibers. Okay, these are the same thing as muscle cells. Okay, so a muscle fiber is a muscle cell. So sometimes you hear people talk about slow twitch and fast twitch fibers, or you talk about aerobic or anaerobic fibers, and what they're talking about is the cells, the muscle cells themselves. And they are covered by what is called a sarcolemma. And so the sarcolemma, sarco, right, is the plasma membrane of the cell. Right, so it's just a special name for the plasma membrane of a skeletal muscle cell in particular. Okay. Now we have inside the muscle cells what are called myofibrils, and myofibrils are organelles. So these are organelles that contain the myofilaments. Okay, so myofibrils, like the mitochondria, right, is an organelle. Like the sarcoplasmic reticulum, which is an organelle, myofibrils are a membrane-bound structure, and they make up most of the space in the muscle cell. So they kind of fill the entire thing, right? So the myofilaments are proteins. So these are contractile proteins. And you may have heard of them before because they have the name myosin and actin. And we'll talk about how they are arranged such that we can get a shortening of an individual muscle cell, and hence a shortening of the whole muscle itself. Okay, so those are myofilaments. We also have another organelle called a sarcoplasmic reticulum. So this is a special type of endoplasmic reticulum, and it stores specifically calcium ions. Okay. And it releases those calcium ions when it is stimulated by a motor neuron. So calcium plays a major role in muscle contractions. So you have to regulate your calcium levels in your interstitial fluid, the fluid surrounding the muscle cells, and as well as inside the muscle cells. Okay. So if we look at the diagram that I've given you, it looks something like this. And so this is just a bundle of muscle fibers. And so when we look at the way that the muscles are arranged, muscles only pull, they do not push. And so they're arranged in these bundles of muscle fibers that will contract, and they would allow the muscle to pull on the bone and causing movement. So for example, the biceps muscle cannot push my forearm down. It can only work by contracting and bringing my forearm up, so in forearm flexion. So what is the muscle that works antagonistically to the biceps to extend my arm? Does anybody know? Triceps, right? And so their arrangements of those muscle fibers determine the plane over which the muscle will contract, right? And so we have oblique muscles, right? So the oblique muscles actually kind of allow for side bending, and they run in kind of a cross fashion, right? My rectus abdominis muscles run this direction, and so they would allow me to flex my torso. Okay, so here you can see the myofibrils, and the myofibrils are in one muscle fiber cell. So this is a muscle fiber cell. So the other thing you need to know about these is that muscle fibers are multinucleated. And so they're formed when embryonic cells, stem cells, fuse. And the nucleus, you can see here, is on the edge, outer edge of the cell. And then the myofibrils, which are inside the cell, make up most of that space. There are also mitochondria, okay? So they can have many mitochondria. And the mitochondria are organelles that are responsible for the production of ATP. So I'll put that produce ATP. And ATP is a common energy currency in the cell, and it is what muscles use to contract. Okay. So if we look at the arrangement, we'll talk about this in a second, but if you look at that there's striations in the skeletal muscle, and the striations are due to the arrangement of the actin and myosin. So you notice that the actin here is thinner than the myosin filaments, which are located right here. So, oh, they have it labeled here. So these are the actin, these are the thin. So I'll put thin filaments are the actin. You actually already have this written. And thick filaments are the myosin. Okay. Come back to that in a second. Okay. So it was once thought that the filaments actually contracted themselves. So the proteins, it was thought that the proteins could be long, and when the proteins contracted, they caused the muscle to shorten. But that was disproven in like the 1960s. And so this is what is called the sliding filament model. So it means that the filaments do not shorten themselves, but the shortening is due to the sliding of them in relationship to one another. And so I'm going to draw a little diagram here that shows the arrangement. And you have this on your paper, so you might not want to write it. Okay. These thin filaments are what are called actin. So I'm going to write, these are the actin. These are the Z lines. Okay, so these are also proteins. Okay, one of these units is called a sarcomere. So I think that's there, that's on there too. So a sarcomere is just one unit of contraction, right? So it's the space between two Z lines. So then when I look at my myosin, the myosin has something really interesting on it. The myosin has these structures that look like this. And these are called myosin heads. And I'll just draw them on this one. So these are the myosin, and these are my myosin heads. So this is myosin. And these are myosin with myosin heads. So the way that this works is that these myosin heads can form a cross bridge with binding sites on the actin. So these heads can bind to receptors on the actin, and what causes the shortening of the muscle is the sliding of the myosin and actin filaments relative to one another. So when the muscle contracts, the sarcomere shortens. Or maybe opposite. When sarcomere shortens due to sliding, right, of filaments, the muscle will contract. The whole muscle will contract. So we're going to talk more about this mechanism in a minute. But if you look at, in your book, there are some images that are similar to this that show the difference between a sarcomere that's not contracted and one that is. So you don't need to know the bands. So I'm not going to make you memorize I band, A band, M line. Okay, you just need to know the Z line and the actin and the myosin. So this would be one where they are not contracted, right? So they're relatively long. So your muscle actually has a optimal length to it. So if you overstretch the muscle, then the binding sites, the binding cannot occur. But if the muscle is too tight, then it cannot contract either. So your muscle needs to be of the proper length. And then what happens is that the cross bridges form, and then that little myosin head pivots, and it causes the sliding of the filaments together. And so here you can see that this sarcomere is shorter, right, than this one. So the muscle has shortened. Okay. Okay, so that's the sliding filament model. So let's look at how this happens and the different roles of different substances. Okay. So ATP functions to energize the myosin head. Right. So it does this by taking, you can break down ATP, right? And you can turn it into ADP plus a phosphate group plus energy, right? And so at this point, when the ATP has been broken down, the head actually stores the energy, and then it uses the energy from this chemical conversion to pivot, okay? So this energy is stored in the head. I'll put myosin head. And allows it to pivot. So we're going from chemical energy to mechanical energy. And anytime we do that, anytime we convert from one form of energy into another, it's really inefficient. So we release heat, right? So that's why when we work out, when we exercise, we get hot because this chemical conversion from chemical energy to mechanical energy releases as its byproduct heat, and we have to get rid of that through sweating. Okay. So one of the things that's interesting about this ATP is that it must bind to the myosin head to allow it to release from the actin. So I'll put binding of new ATP molecule allows the myosin head to unbind from actin. Okay. So it's really important that ATP is present, otherwise the muscles stay contracted. And this is really actually the basis for what happens when you die. You can go into what is called rigor mortis. And this is sustained a sustained skeletal muscle contraction after death, right? So rigor is contraction of the skeletal muscle after death. And so what happens is that you run out of ATP. So ATP runs out after death. And so it depends upon the circumstances, but you could stay for hours kind of contracted up, right? And then once the cells start to decompose, right, the body starts to decompose, then the muscles get soft again, right? But this is the chemical basis for rigor mortis, okay? So if we look in your book has some wonderful, good diagrams that shows this. So I would recommend studying your book, okay? So this just shows the ATP. So my myosin head, this is already hydrolyzed ATP, and so it is energized, right? And so what happens is that then it binds to actin, and then notice what it does, it pivots. And then it releases this old ADP, and that gets reconverted by the mitochondria. So the mitochondria can produce more ATP from these components. You have to put energy into it to do so, right? And then in order for it to unbind, ATP has to bind again. Okay, so it unbinds by adding the new molecule of ATP. Okay. So you'll notice here that there is another molecule that is important in determining whether or not contraction will occur, and this is calcium. So we're going to look at what happens when your motor neuron stimulates a skeletal muscle to contract. Okay, so we're assuming that there's lots of ATP present in the mitochondria, in the skeletal muscle. Okay, so this is what we call the regulation of muscle contraction, and this is described really well in your textbook as well. So I'm just going to label the steps that you need to know. So 1, the motor neuron releases acetylcholine. We talked about that. That is the type of neurotransmitter that's also used by the parasympathetic nervous system, but it's also used by the somatic nervous system. So the motor neuron releases acetylcholine into synapse with skeletal muscle fiber. Okay. So they actually create a connection between the nervous system and the skeletal muscle. Okay. And acetylcholine is rapidly broken down. So your muscles do not stay contracted unless you keep sending action potentials to them. Okay. The second thing that happens is that ACH, so I'll just abbreviate it as ACH, binds to receptors on muscle fiber. And this causes sodium ion channels to open. So this is kind of a review, right? We talked about how this is an excitatory neurotransmitter, And excitatory neurotransmitters always open up sodium ion channels because sodium ion channels flood in, right? So sodium floods in. And this has a name. This is called depolarization. So whereas neurons can depolarize, so can your skeletal muscle fibers. Same thing. So depolarization, remember, it goes from a positive charge, or a negative charge, sorry, a negative charge towards a positive charge. Okay. So I'd say acetylcholine is an excitatory neurotransmitter. Okay. The depolarization causes calcium to be released by the sarcoplasmic reticulum. So depolarization causes calcium to be released by the sarcoplasmic reticulum. Okay. Remember, calcium also played a role in releasing neurotransmitters into the synapse. Remember, we talked about that role of calcium causing neurotransmitter release. But in this case, we're talking about it going to cause muscles to contract. And so the significance of this is that calcium ions, and it has two positives, bind to troponin. Okay, this is another protein. This is a regulatory protein. So this is a protein. Okay. So troponin is a name of a protein that binds the calcium, and this causes tropomyosin to move. So it causes another protein to move away from myosin binding sites on the actin. I'll just put on actin. So this opens up the binding sites, right? And so this ultimately leads to what is called cross-bridge formation. Okay, so the cross-bridge is where the myosin head binds to the actin. So that's called a cross-bridge. Then the myosin heads pivot, the actin and the myosin slide, and then the sarcomere shortens. And that shortening of the sarcomere down the length of the muscle fiber can cause the whole muscle to contract. So like when I just contract my muscle a little bit, but not really hard, that is only stimulating a few muscle fibers. A few muscle cells are contracting. But then when you recruit more muscle fibers and you contract as hard as you can, then the whole muscle can contract and it can be more, it can increase in strength. Okay. So if we go back to that diagram from your book, this shows tropomyosin, and it is covering the yellow. So the yellow here underneath is actually, it's not coloring yellow. Is tropomyosin the yellow? This is kind of a weird diagram. Actin is the blue stuff, maybe the, okay, so here's a tropomyosin. So calcium can bind, calcium is binding to the troponin, yeah, that's right. So calcium binds to the troponin, and these are the little binding sites, right? And so the binding sites are open. And then this is the cross bridge, okay? So let me see if I have a better picture that shows when it's not. Let's see, okay, here it is, okay. So this is a diagram that shows everything I just talked about, okay? Acetylcholine is released from the motor neuron. It crosses the synapse, it binds the receptors. This is the depolarization, and it goes down into the sarcoplasmic reticulum. So this represents the sarcoplasmic reticulum. So if it is not being stimulated, calcium gets taken back out of the cytoplasm and it's removed, right? And so here you can see the yellow. So yes, the yellow is the tropomyosin. And notice how there can be no cross-bridge formation here. Okay. When calcium is present, notice how the yellow moves, right? And we can get cross-bridge forming. Okay. So this is why it's really important to have calcium in your body, right? We store it in our bones, and we can take it out of the bones if necessary, but calcium in your diet, because you don't want to be hypo or hyperkalemic. But hypokalemic would mean that your muscles aren't going to work very well. Okay, so calcium plays a role in muscle contraction. Any questions about that idea? Sliding filament model. So these guys won the Nobel Prize for their model in the 60s, demonstrating how it works. So it's actually quite amazing what is happening, what is going on inside the cell every time you decide to contract your muscle. Right? This is what is occurring. Okay. So we are going to talk and go to our next topic, which the last topic we're going to talk about this quarter is the reproductive system. Okay, so let me go to File, Open. And you have in your possession also on the back, these are the diagrams for the male reproductive system and the female reproductive system. So we're going to talk about this, and I want you to label them as well. Sorry, I forgot to import. Take a couple minutes here. Okay, so when we talk about reproduction, it's kind of interesting because between sexual reproduction and asexual reproduction, you realize that asexual reproduction is probably a lot easier, and it also would take less energy, and it also would probably prevent perhaps the passing of infection between individuals. So asexual reproduction is without sex. So anytime you see an A in front of a word, right? So can anybody give me an example of asexual reproduction that we've already talked about in class? Planaria, yep. And what? Nope, that is still sexual. Yep. Jellyfish can produce egg and sperm. What does the hydra do as a form of asexual reproduction? It buds, right? So asexual means without sex. So this would be without egg and sperm. So it means that the offspring are genetically identical to the parents. So we'll put asexual. Okay. So does not involve the production of egg and sperm. And the offspring are identical, genetically identical to parent. Offsprings are. Okay, so an example would be fission in the planaria. So we cut the planaria in half, but once they get beyond a certain size, they actually will pull themselves in half. So they actually use their muscular pharynx, which is in the center of their body, to hold onto the substrate, and they'll pull themselves apart, and then you get two, right? We also see this in sea anemones. So sea anemones can pull themselves apart to produce two sea anemones. So this is an example. Does anybody remember what phylum the sea anemone is in? It is in Nidaria. Does anybody remember what class it is in? Anthozoa. Excellent. Awesome. Okay. So they're flower-like. Is this a polyp or a medusa? Polyp, right? So they only have polyps. So they're like coral in that they only have polyps. And so when you go to the coast and you see this group of sea anemones in the tide pool, they're probably most likely a cloned population. So they're probably all genetically identical. So they're a population of clones. Okay. They can also reproduce sexually, but when they do so, their larvae move away from the parents to settle down and to colonize a new environment. Okay. So one thing that's interesting about sexual reproduction, okay, this involves egg and sperm. Offspring are genetically different than the parent. And often the larvae are mobile. So mobile larvae move away from parents. And colonize new environments. Okay, so that would be with the hydra, for example. They can bud, right? So they could produce a whole other individual, but the bud just falls down right next to the original individual. And that makes sense because if I'm doing really good in my environment, good enough to reproduce, I could produce something that's just identical to me and it would do well in the environment. But if I'm going to produce sexually, maybe my offspring aren't going to do so well in my environment, so they need to go out and find a new environment, a novel environment. Okay. So what this means is that things like being hermaphroditic, producing egg and sperm, is still sexual. And even if you have self-fertilization, so we talked about one of the adaptations to being an endoparasite is that you're hermaphroditic and you can have self-fertilization, right? That is still sexual reproduction. Okay, and that means is that the genes, even within me, say if I could produce egg and sperm, they both came from me, but if you remember from your previous biology, the genes get shuffled during meiosis. Okay, so they have what is called crossing over. And so the eggs that I produce are going to be genetically different. The sperm that I produce are going to be genetically different. So even if they come together, those offspring are going to be different than me. Does that make sense to everybody? Okay. So the question is, if we think about the costs and benefits of sexual reproduction, one, the cost of sexual reproduction is that you have to find a mate. Okay. So we'll look at cost-benefit analysis. Of sexual reproduction, okay? So if we look at the costs, okay, you have to find a mate unless you're going to self-fertilize, right? Unless you can self-fertilize. Okay. Right. And then you have to risk getting possible infections from another individual. So sexual reproduction can be risky. And you remember spiders, being carnivorous, evolved a way to reduce that risk, right? The males use their pedipalps instead of their penises in order to fertilize the female, and that helps to reduce their risk of being eaten. So you could find a mate, and infections are being eaten or damaged by your sexual partner. Okay, so this is an example of hermaphrodites, and does anybody know what phylum this organism is in? What does it look like? Sea slugs, and so what phylum are they in? Mollusca, phylum Mollusca, class Gastropoda or Gastropoda, right? So they are related to terrestrial slugs, but also they are related to snails, okay? And so here you can see that they're just exchanging their sperm, right? They just exchange the sperm, and they fertilize the eggs with the sperm that they get from another individual. So they're both male and female. So hermaphrodites, right, are still sexual. So you probably already have that in your book that we talked about what hermaphrodite is. Okay, so then what is the benefit? Genetic variation. Right? So this is particularly significant when we're talking about evolving in response to microorganisms in your environment, because microbes can evolve really fast. And so it's thought that with genetic variation produced via sexual reproduction, that this actually aids in being able to fight off infection by microorganisms. So I'm going to show you a little video that talks about this, and also talks about some examples of organisms that went from being sexually reproductive to like a whole species that then became asexual. And it talks about the possible interesting consequences of that. Okay, see if I can get it over there.
Here in Southwest Texas, roundups have been a big part of history. I mean, we have cattle roundups. Lately, we have rattlesnake roundups, but my crew is not here to look for cattle or rattlesnakes. We're here to look for a particular species of lizard that has a different reproductive strategy than most organisms that live on this earth. Hey, look, there's one.
Jerry Johnson and his students are after a species made up only of females, which give birth without having had sex. Each egg contains a complete set of its mother's genes. It develops and grows without any contribution from a male. And so each baby lizard is an exact copy, a clone of its mother.
Some people think that they actually have to have some kind of a lesbian behavior where a female mounts a female to get the eggs to develop. That hasn't been really proven yet, but it's an interesting hypothesis.
Regardless of how they do it, these lizards have mastered the art of cloning, which raises a fundamental question.
As far as these lizards go, the big question, I think, is they do so well as all-female species. Why is there sex? I mean, are males really necessary?
Immersed as we all are in a culture that extols male courage, grace, self-confidence, passion, questioning the necessity of males is rare. Men almost never do it, and women do it most often in a fit of pique. Most of the time, we're happy to say, vive la différence, and get on with things. But for evolutionary biologists, the question is real. Given the efficiency of cloning, why would any female put up with the complications of sexual reproduction? For starters, males can't bear offspring and rarely share the burden of raising them. Then there's the fact she only gets to pass on 50% of her genes. Not to mention over time, energy, and bother involved in courting and mating.
There has to be some fundamental biological evolutionary reason for sexual reproduction. This has been one of the major questions in biology for a very long time.
For 25 years, Robert Reinhook has been returning to the remote hills of Sonora, Mexico, hoping to find clues to the enduring mystery. Why sex? In this dry and forbidding landscape, he seeks those clues in the lives of fish.
To study the sexual process, which appears to be normal, predominant in most things, I study the diseases of sex, the pathologies of it. Little fish, in this case, that live in the Mexican deserts that have abandoned the sexual process. And the beauty of this is it's a natural experiment, because side by side in these same little puddles, we'll have sexual reproducers, we'll have asexual reproducers, all competing in these tiny little simple environments. I could try to study this in a massive environment like an African savanna, but I can't manage an African savanna. With my capabilities, I can manage a few small pools like this. And for 30 years, I've been coming back to this part of Mexico to study what goes on in these pools. We got some. This is a good sample here. Little blackfish fish are. Wow, what a penguin.
Early on, Reinhook discovered that 40% of the minnows in these pools were heavily infected with a parasite that causes black spot disease. The rest seemed relatively unaffected.
When we brought them back to the laboratory and started counting the spots, we noticed, well, my goodness, this is really neat. The asexual reproducers were taking much higher loads of parasites than the sexual reproducers on average. Why should they be more parasitized than the sexual reproducers? They were living right beside, because they're all being exposed to the same parasites in the waters. We know fundamental reason for different parasite loads on these different kinds of animals, unless it had something fundamentally to do with being asexual as versus being sexual.
But what? The only thing to do was to keep walking and sampling and thinking. Finally, it hit him. He was looking at a real-world demonstration of the value of males, one suggested by an evolutionary theory called the Red Queen.
The Red Queen is an elegant idea. I think it's one of the great ideas since Darwin. And it goes back to a wonderful scientist, Lee Van Balen, who basically asked about, does evolution stop when things get perfectly well adapted to their environment? He said, of course not. Evolution is a race, and it's much like the race we saw in Alice and the Red Queen and Alice in Wonderland, where they were running just as fast as they can. And Alice says, isn't this curious? As fast as we run, nothing seems to change. We're staying in the same place. And the Red Queen says, yes, you have to run just as fast as you can to stay in the same place. You're living in a complex world, a world full of parasites, a world full of viruses and bacteria, predators, competitive species, all basically evolving too. And the moment any species stops evolving in response to these challenges and threats, it's doomed.
The cloned fish have stopped evolving. They're an easy target, especially for a short-lived, fast-evolving parasite. But the sexual fish are a moving target. Each is a new combination fashioned from its parent's genes.
So as far as the parasite is concerned, every individual in that sexual population is a unique and individual challenge. And that slows down the transmission of the parasite through the sexual part of the population.
Here was a solution to the mystery of sex. It's the best defense against rapidly evolving enemies. Or so it seemed, until a bad drought dried up the pools, killing the minnows and throwing everything into question. Eventually the water returned and so did the minnows, having slowly worked their way back up the hill from pool to pool. When Reinhold checked the top pool, he made a disturbing discovery. Now the parasites were decimating the sexual fish. The clones were doing quite well, thank you.
This is absolutely contrary to anything we'd seen for many years previous to this. Now it was completely upside down. Something mysterious was going on. Now an opportunity, you see, to do some science. Nature had done an experiment for us. We had to find out what happened, what changed. We collected the fish. We can then examine them carefully, and what we found in this case is that the sexual species, in the process of recolonization, had lost its genetic variation. It had become inbred.
Testing revealed the sexual fish to be clone-like. They now resembled each other genetically. And since they outnumbered the true clones, they were being targeted by the parasites. As a final test, Reinhold conducted a simple experiment.
I went downstream with my bucket into a lower pool where the fish still had genetic variability, and I took some fish from there, put them in my bucket, hiked back up the mountain, and threw them in the pool where the fish had lost variation, and came back a year later just to see what happened. And it's just a wonderful result. This parasite load in the sexuals had dropped right down to the levels it used to be in the past. The asexuals now were taking the brunt of the parasite attacks. And when we examined those sexual fish, we found that we had, in fact, successfully transplanted genetic variation into that pool. And you see, that's precisely the point. That's what sex does. Sex generates variability among offspring. And when you take that away from a sexual reproducer by inbreeding them, cloning them, you've lost the very benefit of sex. It's that generation of an immense amount of diversity. That diversity of your offspring, it provides challenges to everything around it, challenges to the parasites, challenges to the viruses, challenges to your competitors. That's the beauty of the sexual process, is the variation and wonderful diversity it creates.
Okay, so the Red Queen hypothesis is an example of co-evolution. So what was the other example of co-evolution that we talked about previously? Does anybody remember? That was the snake, the garter snakes, and the rough-skinned newts, right? And so that was that idea that they have genetic variation, right? And then the variation was the amount of toxicity that the newt would produce. And in response, you had the garter snake evolving the ability to withstand that toxin. And so when we look at that, what is referred to as the Red Queen hypothesis. This is the idea that parasites, for example, and hosts, or predators and prey, co-evolve with one another. So as the prey or the host develops resistance, the parasites or the microorganisms or the viruses evolve a new way, a new strategy, or the predators evolve a new strategy in order to find them, okay? So it's this idea that it just keeps going, okay? So that's the Red Queen hypothesis. They also made mention of some species where there is only females. And this is what is referred to as parthenogenesis. And so this is the production of eggs that develop into offspring. So the example that they talked about was the whip-tailed lizard. And it is found in the Southwest, like Arizona. And when scientists went down there, they were amazed to find that all of the individuals in the population were female. And how can you tell whether individuals are female or male? Like if you were going to go into a population, how might you tell whether something was male or female? So whether or not they have a copulatory structure, like a penis might be something you'd look for, right? Or also you might look for what size is the gametes, right? So if they're producing large eggs, right, the gamete size, then you would assume that they're all female. So if all individuals can produce eggs, then the idea was that this is all female. The interesting thing about the whip-tailed lizard is that they are believed to have evolved from a, they're asexual now, they do parthenogenesis, but they're thought to evolve from a population that had both males and females. Okay, so evolved from a population, both males and females. And the reason why they think this is that courtship appears to be necessary for ovulation. So ovulation is the release of the egg from the ovary, right? And the courtship behavior is they have to have an individual on their back, and in the male courtship, it's they bite the neck of the female. And so that courtship behavior has to be present in order for the females to release their eggs. And so if we look at what happens in a population, it's kind of interesting because these are two females, but this female is not getting ready to ovulate. And so her ovary size is really small. And so she is behaving like a male would in a sexual population. And then this female underneath is getting ready to ovulate. So she is behaving like a female. And so the way that they behave depends upon where they are in their reproductive cycle and where their hormones are. So if estrogen is really low right here, then let's see, what are they showing here? They're showing the red. So this would be a female that is getting ready to ovulate, right? So that individual acts like a female. After she ovulates, she acts like a male, right? And so one of the interesting things about this is that they actually discovered that the offspring are not quite clones because they're producing eggs. So there's very little genetic variation, but the offspring can be slightly different because there's crossing over between homologous chromosomes prior to the production of the egg. And so the egg can be slightly different from the parents, but there's no, new genetic variation being added. Okay, so this is kind of a question mark. Why would a population go from having males and females to simply losing the males entirely and becoming all females? Okay, so we're going to take maybe a 5-minute break here. I'm going to go to the bathroom, and we're going to talk about sex determination and mechanisms of sex determination. You get to ramp up and walk around the device. Thanks.
It's almost like it's like maybe one drink of ice. viewing for my health class, and I cut on with now.
Priorities.
I know, right? Gotta sleep.
There's two C's.
Come back there.
Okay. So when we talk about sex, and we're looking at the differences between males and females, we can talk about biological sex. And this is determined by the size of the gametes. Okay, so gametes would be the egg and the sperm. So we would say the females have large gametes, and the males have small gametes. So sometimes doesn't have to do with like which individual becomes pregnant with the larvae. So for example, a good example is in seahorses, and those are bony fish, and the males actually receive the eggs from the females. So the males actually become pregnant, right? And they get, their babies grow and develop in a pouch on their belly, and then they give birth to live young. So it's not even which ones provide most of the care or which ones receive the gametes. It seems to be based upon the size of the gametes. But because females tend to produce or put more energy in most cases towards reproduction, the females tend to be kind of the limiting resource. And so if you look at sexual reproduction in mammals, the females generally always get to reproduce. They have to expend more energy and time in caring for the offspring, but there are males out there that have to compete, right? And so maybe only the most dominant males actually get to reproduce, okay? But if you were looking at a population, if you find the individuals are producing large gametes, you would say that they're female, right? And then the smaller gametes would be male. Okay, so in our sex determination, it's genetic, right? And we know that it can be determined by chromosomes, but some organisms have environmental or determination. So this is as opposed to versus genetic. So this is not the way we do it, okay? When we look at reptiles, one way that they sometimes regulate their sex determination is by temperature. So the temperature of incubation, right? So it could be, and sometimes it's opposite, but it could be that the warmer that the eggs are incubated at a warmer temperature, they become female, and at a cooler temperature, they become male. So it has nothing to do with genetics. The male and the female are genetically identical. So it just has to do with the environment, okay? So this is an example, okay? Another example would be what other individuals in the population are doing. So your environment could be what other individuals in the population are doing. Doesn't make sense. I'll just put other individuals in the population. That's my environmental. Other individuals in the population. Okay. So for example, fish sometimes have a mechanism called sequential hermaphrodite. So a sequential hermaphrodism, can't spell that right. Ditism, there we go. Okay. So this means that they can be born a female, and then later in their life, they turn into a male. Or they're born male, and later in their life, they turn into a female. And so, for example, in clownfish, so this would be like Nemo, right? In Finding Nemo, right? Yeah. So clownfish, if he loses the female, he could turn into a female. So Nemo could have become a female, right? So if you take a bunch of clownfish and you put them and you isolate them and they're all males, then one of them, usually the most dominant, will become the female, right? So it depends upon what other individuals are doing in the population, what sex you are, right? So that's called sequential hermaphrodism. Okay, so this is environment. We can also look at genetics. And one example that's really interesting is what is called haplodiploid sex determination. So if you remember back in genetics, haploid means half. And so this would mean that they have only half the genetic information as somebody that is diploid. So a good example of this is social insects, like termites, like ants, like bees, okay? So for example, bees, my example. Okay. So in this particular instance, the male is haploid. And the females are diploid. And so who are the workers? Females are the workers, right? So those honeybees that you see flying around are female. The males, there's very few males, and the males fertilize the queen, right? So when we look at, these are the workers, so they don't get to reproduce. So they're all sisters, right? The female queen, right, is also diploid, right? She produces eggs that are then fertilized by the male's sperm, right? And the queen is queen because of the environment and specifically her diet. So this is due to being fed what is called royal jelly. So when they lay an egg into a particular section of the colony, and if they put royal jelly in with the egg, then the larvae eat the royal jelly, and that affects the gene expression, and then that female turns into a queen, right? And so it's the queen that gets to reproduce, not the workers, okay? But the workings, I should have put workers, sorry, the workers, okay? But the queen and the workers are very closely related to one another, right? And so, especially the workers are like, 2/3 of their genetic, they share about 2/3 of their genetic information. And so that is one way that it kind of keeps them in a environment where they're very social, because they aid in the production of sisters that are actually more closely related than we would be if we were sisters. Okay, so I wanted to show you a little video of that idea of haploid diploid. So I forgot to put it on here though. Find my science crash course. Oops.
So well, in this video, I'm going to explain how
This
to
isn't the
deploy
video I
the
wanted,
system
sorry.
on,
I can't
honeybees
find it right now.
and honeybees.
Okay, sorry, I had a good video and I just can't find it at the moment. Okay, so the interesting thing about this royal jelly is this shows how nutrition can influence gene expression, right? So royal jelly is really interesting because of that. It's called nutrigenomics, is the study of how nutrition can influence the genetic expression. So this is nutrigenomics, brand new area of research. And so this is how food can determine gene expression. Okay, so we have sexual determination via genetics as well. And so we have chromosomal differences. So males are what combination of sex chromosome? Yes, so XY. So they are said to be the heterogametic sex. And so you might hear, oh, it's the male that determines the sex of the offspring. And it's true because it's the sperm, right? So the sperm can either have an X chromosome or they can have a Y chromosome. So hetero means other gamete. So this means that they can produce one gamete over the other, right? And XX is the female, and this is the homogametic sex. So the female can only produce eggs with the X chromosome. Okay, and so So one of the really interesting things is that this presence of this Y chromosome actually makes it so the sperm with the Y chromosome are actually faster than the sperm with the X chromosome. And so more boys in general are conceived than girls. However, boys have a higher mortality. So by the time they get to reproduction, the sex ratio tends to be 1 to 1. So for every 1 female, there is 1 male. And so I wanted to show you a short video that kind of goes fast into, it's kind of a fast video, but it talks about sex ratios in humans and when they become skewed and under what conditions they become skewed. So.
From our home in New Orleans to your kitchen, possibilities are cooking. It just takes an idea and a little inspiration from Zatarans. With more
Just
than
ignore.
20 flavors.
Don't listen to it.
Wrap it up. Most parents-to-be assume that the sex of their child comes down to a flip of the chromosomal coin, a boy or a girl. But in reality, the odds aren't even. For every 100 girls born, the world gains about 106 baby boys. Currently, that skewed sex ratio comes out to roughly 10 million more baby boys than girls born worldwide each year. There are some countries where human meddling stacks the odds even higher in favor of boys, but that doesn't explain the fixed odds everywhere else. The intrinsic boy-girl ratio is rigged by Mother Nature. In fact, it's even more rigged than birth rates show. Human conception results in about 150 male zygotes for every 100 females. But there's a rather tragic reason for this big biological boy bias early on. Male fetuses are much more likely to be miscarried or stillborn than female fetuses. And boys that do make it out of the womb suffer more fatal diseases, take more mortal risks, and fall prey to more violence than girls. So by the time kids grow up and reach baby-making age, the ratio of males to females is just about 1 to 1. But the likelihood of a boy even making it to birth is also influenced by his mom's living conditions during pregnancy. For example, when a massive famine struck China in the 1960s, the relative likelihood of having a son suddenly dropped until the famine ended. And male Americans born to billionaires seem to have higher than average odds of fathering sons. Somehow, female biology suppresses boy survival in the womb during tough times and boosts it when times are good. We've seen the same pattern in other mammals too. When resources are scarce, mothers give birth to fewer males than normal. When resources are plentiful, they bear more. The best explanation we have for this has to do with sex, the other kind. In biological terms, the whole goal of copulation is to reproduce, to pass down your genes to someone who will someday pass them on again. Female offspring are almost guaranteed to reproduce, famine or no famine, because male mammals are pretty much always willing to mate. Males, on the other hand, have to compete for mating privileges. A well-nurtured hunk has a good chance of mating with lots of females, while a male weakened by famine might not score at all. So male offspring are a bigger risk in general. At all stages, they're more likely to die. And even if they live, they might not reproduce. But when times are good, boys' potential to father lots and lots of babies make them a biological risk worth taking. Thanks to everyone who supports us on Subbable. Without you, we literally couldn't.
So the sperm with the Y chromosome are faster, so that more baby boys are conceived. But remember that they die at a higher rate. And it also depends upon the mother's environment as to whether or not she will be receptive to boys. So if her environment is really bad, like in a famine, then the boy babies tend to die and be miscarried at a greater rate than females. Okay, so you could say that there's chromosomal differences, and there's generally a 50% chance of having a boy or a girl, but that also depends upon the environment. Okay, so our chromosomal differences are not always this way in all vertebrates. So if we look at birds, it's exactly the opposite. So in birds, instead of an X and a Y, they have a Z and a W mechanism. So ZW chromosomes, that's their sex chromosomes. So the female is homogametic. She is ZZ. Oh, sorry, heterogametic. She's ZW. It's opposite. ZW. and then the male is ZZ, right? So in this particular instance, it's the female that determines the sex of the offspring. Right? So it's whether or not the egg has a Z or a W, and then you would predict that there would be more even skewing of the sex ratios because there's not a faster or a slower sperm. Okay, they would all be the same, pretty much the same speed because they are carrying the same amount of genetic information. Okay, so those are just some different mechanisms of genetic biological sex determination. And so we're going to start today talking about male reproductive structures. Okay. So when we look at the gonad, these are the testes. So the gonads are where meiosis occurs, which is a special type of cell division that produces haploid cells. And in this case, the haploid cells are sperm. So this is where the production of sperm, so this is, we'll put sperm are produced. Sperm production. Okay. The testes are actually located in mammals in outside of the body cavity. So it wouldn't be the same thing with fish. Fish, the males have the testes up in their body cavity. But when we look at endothermic organisms, so endotherm, specifically endothermic mammals, have a scrotum. And the function of the scrotum is not to protect the testes. That's what always people I say, what's the function of the scrotum? Oh, let's protect the testes. But think about it, compared to the ovaries, it's not protected at all, right? I don't see that the testes or the scrotum protects them very well. But what it does is it regulates the temperature at which sperm are produced. So this is temperature regulation. So for some reason, and I don't know why, sperm have to be produced at about a -3 degrees Celsius lower than your center of the body. So if the testes were up in the body cavity, the sperm would be defective, right? And so pulling the testes up close to the body keeps them warm, and then when the body gets too hot, the testes fall away, right? So this is temperature regulation, and that's important for proper sperm production. So they always say if you're trying to get a female pregnant, like if you're a couple, that males should not wear tight underwear, right? Because that tight underwear might keep their testes too hot, and then the sperm get mutated, and then they can't fertilize the egg. Okay. So that's the testes and the scrotum. Okay, so we have what is called the vas deferens. Actually, sorry, I need to mention the epididymis. Epididymis. That's not epididymis. Oh, I spelled that wrong. Okay. This stores sperm prior to ejaculation. Now, there does seem to be a shelf life to sperm, so that if they're stored too long, they become what is called sticky sperm. And so sticky sperm are not able to fertilize the egg as well as non-sticky sperm. And so the male needs to get rid of the old sperm and get new sperm in order to help increases the likelihood of fertilization. Okay. So that's the epididymis. And then we have the vas deferens. Sometimes this is called the ductus deferens, but we'll just call it the vas deferens. And this is the duct that transports the sperm up into the body cavity upon ejaculation. Because that is where the glands are located that are going to add substances to the ejaculate. Okay, so the sperm are produced outside the body cavity, but then they have to go into the body cavity to pick stuff up. And so we have what are called accessory glands. And these add substances to the ejaculate. And the two glands include the seminal vesicles, which there's two of them, seminal vesicles. So there's two, okay? And the seminal vesicles particularly add, there's some other things that they add, but they add fructose. And so fructose is what is used by the sperm as energy, which is really odd to me. Because if you think about it, that's fruit sugar. So if you think about, you know, high fructose corn syrup, how bad that is for our livers, right? And we usually, as animals, we don't use fructose. We usually use glucose, right? But fructose is what the sperm need to eat in order to produce energy that allows them to swim, okay? So it is fructose. They also produce a coagulating enzyme. Okay, so this is energy. Okay, this actually helps to keep the sperm, when it is ejaculated into the female, the sperm wants to coagulate and so that it doesn't run right back out of the female. So like if you think about the female reproductive system, the sperm could just flow back out. And so the coagulating enzyme solidifies the ejaculate. Upon entry into the vagina. So you don't get what is called backflow out of the reproductive tract. Okay. We also have the prostate gland. And there's only one of these. Okay. But the prostate gland also adds, kind of interestingly, an anticoagulating enzyme. To become thinner so that the sperm can swim. Oops, ejaculate. Okay, and the sperm actually have to swim up through the female's vagina, up through the cervix, up through the uterus, and into what is sometimes referred to as the fallopian tubes, where the fertilization takes place. So they have to go a long distance, right? So they need lots of energy. They need lots of energy and nutrients. Okay. So if we look at our diagram, so if you want to look at your diagram that I gave you, okay, we'll label some of these, right? So this is my scrotum or scrotal sac, right? This is the testes. This right here is my epididymis. Okay. Now this is really confusing because it looks like there's just a tiny little duct that travels by itself, but really this vas deferens, this duct, is actually in a bunch of muscle and nerves and blood vessels, and it's called the, there's a name, oh, I can't remember what it's called, but this is not, they just took away all that stuff, right? So there's actually a lot of other stuff that travels with that. So this is my vas deferens. So if you are going to have a vasectomy, they would go into that bundle of tissue and they would just cut the vas deferens so that the ejaculate would happen. You would still have stuff from this and from the prostate glands, but you wouldn't have any sperm in the ejaculate, hopefully, if you're trying to use that as a form of birth control. Okay, so this right here is my seminal vesicle. And then this is the prostate gland. Now there's one more gland that actually doesn't add stuff to the ejaculate per se, but it produces what is called pre-seminal fluid. And so this is this structure right here. So what this is called is the bulbal urethral gland. And it produces pre-seminal fluid. And this has a couple of functions. You'll notice that its secretions would go into the urethra. And you know that the urethra is part of the urinary system, which carries urine. And so if you get pre-seminal fluid in there, it removes the urine. And that actually is a really good thing, specifically because the urine can be very acidic and it could kill the sperm, right? So this kind of removes the urine, and it also functions in lubricating the penis. So it removes urine and lubricates the penis. Now, some people get concerned because they say that there can be sperm in the pre-seminal fluid, but there wouldn't be sperm unless the male had ejaculated recently, because the sperm can live for a couple of days maybe in the reproductive tract, right, of the male, but they wouldn't be able to live because of the presence of urine. So when the male urinates, that probably kills the sperm, I would imagine. So it would only be like if the male had ejaculated recently and had not urinated, that there might be some leftover sperm in the urethra, which could lead to an unwanted pregnancy. Okay, so that's the bulbal urethral gland. So you'll also notice the prostate gland here has part of the urethra coming through it. So the prostate gland is notorious, specifically in our culture, because for some reason, white males in the United States, if you live past the age of like 70, you're almost guaranteed to have prostate problems. And so some people think it's genetics, some people think it's environmental, like what we eat, but this becomes enlarged and then it becomes very difficult to urinate. And so males with prostate gland issues, and it can turn cancerous, but sometimes they go in and they actually remove stuff here. So they'll go in and they'll take out and make this duct a little bit bigger so that the male can urinate and kind of remove some of the prostate so then the male can urinate. That does not become a problem. Okay, so I'll stop there for today. And so you want to bring your handout to class on Monday, and we'll finish talking about male, and then we'll talk about the female reproductive system on Monday. So Monday at noon. But I will see you tomorrow at 2 as well.